Hi, I'm Cameron Burke, and this is the Kaplan MCAT Science Review for the Fluid Mosaic model. This video covers a super broad picture of what's going on with the cell membrane, including the membrane's general functions, a definition of what the fluid mosaic model actually is, and the membrane dynamics of the different molecules that make up the cell membrane.
For this video, the main goal to keep in mind as you work is that you want to establish a general model of the plasma membrane. By the end of this review, this goal should help you answer questions like this one about the role a plasma membrane plays. The most important job a cell membrane has is to protect a cell. Secondarily, it also plays a big role in sustaining and regulating cellular activities. Membranes serve many functions, including creating selective permeability, intermolecular communications, and intermolecular communication, and transport of biologically important molecules both within a cell and around a body. These are just a couple broad categories, but the big picture is that cell membranes have super important responsibilities. The fluid mosaic model helps explain the components of a membrane that let these various tasks get accomplished. The fluid mosaic model is a useful way to think about the components that make up the plasma membrane. The membrane itself is the fluid mosaic, where its components create hydrophilic planes on the inside of cells. And on the outside of cells, while hydrophobic fatty acid tails create a hydrophobic inner region between the inside and outside of the membrane. This differential allows the membrane to effectively regulate what sorts of molecules can pass through the cell and under what conditions the passage can happen. The hydrophilic regions are mostly made up of phosphate heads, proteins, and carbohydrates. The fluid in fluid mosaic model refers to the fact that the components of cell membranes aren't static, but rather that they can move around as needed within relation to each other. Phospholipids in particular align themselves through diffusion to get to the most favorable polarity alignment, with the hydrophilic and hydrophobic regions in the right places. In addition to being arranged by a polarity, they help maintain the density of the cell membrane by moving to areas where they're least concentrated, so no one area has too many phospholipids. Phospholipids are small and have charge, so they get to zip around relatively quickly. But larger molecules like lipid rafts with cholesterols in them and proteins scoot around more slowly. That said, all the components move around to where they need to go. Because the membrane is a fluid, stuff gets shuffled around. Sometimes the concentration gradient of lipids dictates that some lipids need to move to the opposite bilayer of the membrane from the one they're currently in. Flipases are enzymes that help the little lipids get where they need to go. Flipases basically pick up the phospholipid who need to move across the hydrophobic region and turns it over so it's on the opposite side of the bilayer. This way, hydrophobicity isn't a barrier to fluidity of the membrane, and diffusion isn't inhibited. Now that we've covered the content for this review, check out how you're doing with understanding membrane basics with this practice question about the role of the plasma membrane. The correct answer is D. The plasma membrane has nothing to do with breaking down proteins, but it is super important for cell recognition, waste excretion, and material transport. Phospholipids can diffuse very quickly due to small size and charge within one plane of the lipid bilayer. Big proteins and lipid rafts can also diffuse, but due to their size, it happens more slowly for them than for phospholipids. If phospholipids need to get across the membrane, they need a flipase enzyme to help them out.
Thanks so much for watching this review of the fluid mosaic model. We hope it helps you out if you get questions about the fluid mosaic model on test day.